When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
So, in this question, we are going to look at genetic engineering. So, physically, in the question itself, when we go and look at it, it gives you a reasonable amount of information. Right, it's really important that before going and even thinking about the information that it asks you, just try and just run through it in your own head how, or the process that genetic engineering, how it happens. So the first thing that we do when we're genetically engineering anything is that we go and we identify the gene that we're going to be cutting out, whatever. We then go and extract the DNA, so the chromosome, from that particular nucleus of the cell. We use restriction enzymes to go and cut out the specific gene from the cell. That gene is what we're then going to go and use to alter our altered bacteria in this case. So if we're looking at altering bacteria so it produces insulin, we go and we identify as plasmid. We then extract the plasmid from the bacteria. The plasmid is cut open using the same restriction enzymes as we cut out the gene with. And we then go and splice in the genes using a second enzyme, so DNA ligase. That plasmid is then reinserted into the bacteria. So now in this case, the bacteria has the gene to go and produce human insulin. We can then grow the bacteria in a fermenter. As it grows, as it replicates, it produces the insulin which we can go and use and so forth. So if we look at this question in particular. So insects can be useful and harmful to crop plants, right? So that gives you some instant clue about what it's about. So insects such as bees pollinate the flowers of some crop plants. Pollination is needed for successful sexual reproduction of crop plants. Some insects eat crop plants and other insects eat the insects that eat the crop plants. So corn borers are insects that eat maize plants. So all this top bit here is all kind of just background information. So that moves on to a toxin produced by the bacterium Bacillus thuringiensis kills insects. So scientists grow this bacteria in large containers. So the toxin is collected from the containers and sprayed over the maize crop to kill the corn borers. Right, so that automatically gives you a clue that it's going to be something to do with removing the gene for this toxin and putting it into the maize. So a company has developed genetically modified maize plants. So GM maize plants contain the gene from the bacteria. This gene changes the GM maize plants so they produce the toxin. So describe how the scientists can transfer the gene from the bacteria to the maize. So think carefully about the range of things that we're going to do that we've just looked at. So the key things that you need to answer this question, you need to talk about cutting the gene out, right? So you're cutting it out of the bacterial chromosome, right? It'd be nice if you can make reference to those restriction enzymes. then talk about the fact that that gene is then spliced into the chromosome of the maize plant. It's an early stage of development, but that will enable it to go and produce the toxin. So if we go and look at a few benefits of genetic engineering, so genetic modification is a lot more fast and gives a lot more predictable results than selective breeding. So it helps improve crop yields. It means that we can introduce herbicide resistance and pesticide resistance without using weed killers. And insects and pests we can discourage them from eating them. So if we look at this question, so it said, what would you advise GM maize plants or farmers to do with it? Right, justify your answer given advantages and disadvantages. So we've talked about the benefits. So the advantages that you would have to talk about is that less effort for the farmer because they don't have to go and keep spraying them with pesticides. You'll have to talk about them not having to go and wash things. Give examples of things like not having to use sprays. Talk about the benefits of the fact that there's less insects to eat the crops or the maize plants or to carry the disease. If you want to talk about the fact that the pesticides don't go and contaminate the watercourses as well, that would also be good. Right. So on top of that, it says that you need to talk about the advantages and the disadvantages. Right. So some of the risks of genetic modification is that transfer from the selected gene that could go and get passed on to other species where we don't want it. Right, some people believe it's not necessarily ethical. So GM crops could be harmful. So for example, toxins from the crops could be detected in someone's blood. GM crops could cause allergic reactions in people. Right, and pollen produced by the plants could be toxic and harm insects if transferred between plants. So some of the key things that you could talk about, the risks and the disadvantages, are things like the toxin kills other insects that we don't want. We could talk about some crops don't get pollinated properly. You could talk about it might be possibly harmful to humans or animals that go and eat them. You could talk about it could damage the food chain, right? And the genes could go and spread to other species. So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.